you should have grabbed this paper on your way in the door. Your copy is green. We're going to go through, this is a graphic organizer. We're going to look at DNA replication, which is old. We learned about that with cell cycle and PMAT, right? And then we're going to compare it to transcription and translation, because I want to be sure you have both and all of these cycles completely separate and clear in your mind. So we're going to start in the DNA replication column. One strategy you could use here, you don't have to, but if it were me, as we change columns, like I'd do my DNA replication notes in one color, and then I'd switch colors to transcription, and then I'd switch colors to translation, just because science says the more visually appealing and the more intentional organizational use of color we use in our notes, the more we remember them. So if you have a class you struggle in, this is just one strategy that you can use, and science backs up with data that it actually does affect how much you can remember. All right, so we're under that DNA replication column. On the left-hand side, the first question is function. What is happening? So during DNA replication, DNA is copied. During DNA replication, DNA is being copied. This is happening in the nucleus. And the purpose of this is to make sure that identical cells are made in mitosis. So we're going to say to make identical cells in mitosis. And you can remember mitosis is PMAT. Prophase, metaphase, anaphase, and telophase. So the process, how's it happening? And it wants us to do a quick drawing. There's not a ton of space here. I'd probably be able to do this a little easier on a piece of paper. But if I did it on a paper, then it's harder to record. So bear with me. I'm going to do my best. If I run out of space in my box, I am going to move to the margin. But y'all remember that you're trying to draw it in your box, okay? So DNA splits. DNA splits, and it adds nucleotides. So I'm going to draw so on the left-hand side, my DNA is not split, right? On the right-hand side, it is split open. So to make this a little clearer to see, I'm going to put my new copies in a different color. So I'm going to come in, and let's say this is wrong thing, Stoley. This was what I had, my original. A, T, A, G. And my original was T, A, T, C. Because you see those match, right? My originals here are complementary. So then I have to come in with my new, and it's going to be the opposite of that. So my new strands would be T, G, T, C, right? And that's on one side. And on the other side, I would have A, T, A, G. So what I've drawn in red is the new copy, right? And what is in black is the original strand. So what I end up with two strands of DNA. One half is old of that new strand, and one half is new of that new strand. It's called being semi-conserved. The next question wants to know what organelles and enzymes are involved. So this is happening, like we said, it's in the nucleus. There's several enzymes here. We've got those 4 superheroes of DNA replication. But the 2 big ones that we talk about a lot are helicase. He's the unzipper. And polymerase. Those are the 2 big enzymes. And the final product are 2 identical strands of DNA. So that's replication happening in the cell cycle, right? What phase? Do y'all remember? S. It's happening in the S phase of the cell cycle. So now let's talk about transcription. I'm going to change my ink color here. I'm going to go to a blue. So transcription, I'm going to circle the C because that helps me know it comes before translation because the letter C comes before the letter L in the alphabet. So the function, what is happening? DNA is being written out into messenger RNA. So I'm just going to say DNA to mRNA. What's the big difference between DNA and RNA? What letter can RNA not have?
R, D, T.
T. It can't have thymine. What is thymine replaced with? U. Uracil. Very good. So this again, this transcription is still happening in the nucleus. And its purpose is to send the DNA instructions out of the nucleus and to a ribosome. So to send instructions out of the nucleus. To send instructions out of the nucleus. Those instructions are your genetic information. The DNA is too big. He can't leave. We've got to get those instructions out somehow so we can use them to build proteins. So then what's the process? It's very similar to replication, isn't it? What's the only thing different? Well, there's 2 things different. I'm using a different base, U instead of T. And is my final product going to have 2 strands or 1 strand? It's only going to be single-stranded, right? So we're going to, the process, I'm going to go back to my black to draw that same strand of DNA, okay? We're going to go A, T, A, G. And then my new strand, my mRNA this time, we're going to put here. It's going to be U, A, U, C, right? And this time, it's going to be a single strand. Not double-stranded like before. We are now at a single strand. The organelles and enzymes involved here, we're still in the nucleus. And we are using polymerase again, but this time it is RNA polymerase. The final product is a single-stranded mRNA that codes for a protein. Product here is a single-stranded mRNA that codes for a protein. Do I need to write that higher up so y'all can see it? So, oh, I don't know what just happened. There it is. That's what's in my bottom box. Single-stranded mRNA that codes for a protein. I'm going to change my color again. I'm going to go to, let's do green. And this time we're talking about translation. So I'm going to circle my L. I circle that L so I can remember that translation comes after transcription, okay? And I can remember that because that's how those letters fall on the alphabet. So the function. In translation, my mRNA is translated to a protein. My mRNA is translated to a protein. Now this is happening someplace different, isn't it? Who knows? It's happening in the cytoplasm on what? It's an organelle. rRNA. It's happening on a ribosome. In the cytoplasm on a ribosome. When you see rRNA, it just means ribosomal RNA. The purpose of this is to make a protein. Whole point of this is to turn that genetic information into a protein. Now we're going to draw it. So I'm going to go back to my blue because I want to show you that RNA strand. It was UAUC. That's what we just made in transcription, right?
Yeah.
So now I'm going to go back to my green. We got lots of steps here. So first thing it's going to bond on, and I don't know why I always draw my ribosomes red. It's going to bond to my ribosome, which is that rRNA. Remember, we talked yesterday about things called codons. Codons are sets of how many letters? 3. There's 3 letters in each codon. So I have one full codon here, right? The UAU, we can say that's a codon. So a tRNA that has the anticodon to that codon. So that means they match. They're complementary. So in this case, it would be AUA, right? Be our anticodon on our, this is tRNA. It kind of looks like a lowercase t to me. And that tRNA molecule has luggage. He's carrying a little monomer. Who can remember what we call those? What does the tRNA carry? He's carrying an amino acid. That amino acid gets dropped on that ribosome, and that tRNA moves, and then the next tRNA comes in that matches the next codon, and that process continues. Those amino acids bond together until they hit a stop codon, which will make a protein. So the organelles involved here, we've got ribosomes, we've got mRNA, we've got tRNA, we've got rRNA, and we've got amino acids. Those are all the key players. And now that bottom left-hand corner on my screen is covered up by the Doceri logo. So I'm going to write what goes in that box kind of slightly above it right here in this area. We'll pretend that's my box, okay? So the final product here is an amino acid chain, which makes a protein. Proteins, if you remember from the very beginning of the year, those are one of our biomolecules, right? They're the one that give muscles their strength. They give, they have the elements CHON, Chon. So give me a fist of 5 on having all of this written down. Those of you who are completing this later in the day on a Chromebook, if you are not done writing this down, you can pause the video at this time until you get it complete and then resume once you are done. I'm going to pause live. Mm-hmm. Is there anyone who needs me to wait? All right, so if you notice the bottom of this page, you've just got one more small section we need to do together, and then we'll be done with this activity. So I'm going to scroll down. And we're going to look at this section here. So this is very similar to what you see on our exit ticket board. We have not made it to that exit ticket this week, which is fine because we're about to do it right now, just in a slightly different way. So we're going to compare and contrast DNA and RNA and their structure. So how many strands does DNA have? It has 2. And if I were to draw a picture of DNA, remember it's that swirly, twirly ladder looking thing, right? And then in the middle are my nitrogenous bases, which are actually my genetic information. The middle is what is important. We have a name for that. We call it a double helix. DNA is made of subunits. Okay, there's pieces that make up a DNA molecule. These are called nucleotides. If you remember back from when we learned about biomolecules, these nucleotides have 3 key pieces. They have a phosphate, they have a sugar, and a nitrogenous base. So this sugar in DNA, he has a name. He is deoxyribose. I'm going to put the letter D on that sugar. This is my phosphate, and this is my nitrogenous base, my 3 parts. So my phosphate and my sugar are the backbone. My nitrogenous base is my genetic information. The bases that can happen in DNA are adenine, thymine, guanine, and cytosine. These are apples and trees and cars and garage, right? And DNA is found in nucleus. Remember, he's too big to leave. He can't fit out of the nuclear pores. And now we're going to talk about RNA. RNA is single-stranded. It just looks like that. It could be mRNA, it could be rRNA, it could be tRNA. That's messenger, ribosomal, and transfer. RNA still has subunits. It still has nucleotides. I am still going to have a phosphate. I am still going to have my sugar. This time his name has changed. It's now ribonucleic acid, not deoxyribo. It's now just ribonucleic acid. And then I still have my nitrogenous bases. But the difference here is I don't have hydrogen at the center bonding one base to another base. It's just a single strand. So there's only one side of backbone and one side of bases. There's nothing on the other side. The bases in my RNA are A, U, G, C. So T's gone now. We can find these in the nucleus, on a ribosome, and in the cytoplasm. Once you get this done, you're going to fold this graphic organizer in half and glue it in your journal hamburger style on the next blank page. At that point, I need you to put up your glue and your highlighters. Once you are finished with this, check the screen at the front of the room for your done activity. There's going to be directions there for what you should do if you finish before the bell.